And our first speaker is Teresa Sholars, who spoke in the morning, and she really was sort of the beginning organizer of this session, and they brought me in, and I ended up being here at the end, so thank you, Teresa, for bringing me in for this. Teresa is Professor Emeritus from the College of the Redwoods. She's very active in botanical communities and does a ton, as many of you know. She's the author of perennial Lupinus, the Jepson Manual, and she's here to talk to us today about practical guidelines for detecting and conserving cryptic species. Oh, we need our first slide.
Good afternoon. Now I take off my ecologist hat and put on my systematist hat, but I will tell you I never really take off my ecologist hat. It's there forever. Practical guidelines for detecting and conserving cryptic species. Well, I first kind of have to say that, you know, my degrees are really in ecology, but I started with looking at lupins, helping Jim Harding back in the 70s when I was at Davis, you know, try to figure out how to get those seed coats open and plant all these ones for his genetic studies. And then, you know, 15 years later when I went back to school at Berkeley, Jim Hickman saying to me, "Gee, I got a great genus for you. How about lupins?" And I thought, oh, I like lupins. I've never kept them before, but I like them. So, wow, you know, so I got sucked into lupins, which are a great genus. Okay. And then there are the species. So, I mean, if there's anything about diversity of species, and cryptic species, and where are the lines between species. I've been thinking about this for a long time with trying to write the perennial lupins for the Jepson Manual, and I just finished, everybody, yay, the Flora of North America. Great book for lupins, so God, it's a mess. Because lots of gray. But Julie and I got asked to speak at the CNPS symposium Aaron Sims asked us, and we actually were the two people who frankly delved in the practical world. You know, I've been a community college professor for 45 years and a botanical consultant, and frankly, I'm just an ecologist at heart, even though I really dabble in systematics. And lupins are a great thing to dabble in. And I found in trying to get a handle on species that I really didn't know as well as I do my California ones, or even some of the California ones, that the literature that I was reading, that people were actually, you know, doing their molecular studies and finding conclusions that and publishing them with not looking at any of the morphology or talking or making any kind of morphological analysis of what was happening, which then makes it very hard for those of us in the field to go in and then how do you tell it apart? Or it's rare, how do we defend that? So, you know, we basically feel pretty strongly that this has got to be integrative sciences where we get people looking at something also besides the molecular end. So now that more and more molecular studies are being done to help, you know, clarify the relationships between species, and some of these studies publish new taxonomies, new classifications, new circumscriptions, without doing species descriptions. In other words, they're saying that this group is one species, this group is another species, but not necessarily, and this is not everybody, but some of them, are doing taxonomies without actually literally describing the species and looking at morphological characters. So this obviously creates a problem for those of us who are in rare plant protection and rare vegetation protection, because we need to know what we're looking at. So we got together, the two of us, and the larger, I would say, community, botanical community. We put that out and got feedback back. And so we have 9 practical guidelines for publishing or recircumscribing new species, especially these groups known as cryptic species. There were 10, and I lumped one together because I'm a lumper, not a splitter. Because I work on lupins, by the way. Anyway, but, this isn't necessarily in any order, but before publishing a cryptic taxon, contact agency or other knowledgeable field botanists who can help you research the subject in the field. So, we need to use our colleagues, and we need to talk to other people who actually might know the group that you're talking to more than you do, or have some guide, some suggestions. So talk to people in CNPS, talk to the people at the Fish and Wildlife and the CNDDB. Please, number 1, before publishing the cryptic taxon, contact them and get the names of people who might be able to offer you help in the field. Maybe helping you do some field surveys if that isn't your skill set, helping identify suitable habitat, or helping you separate some ecological things so that, because this may not be your thing. And issuing permits, by the way, for public lands, and then contact these people, have them help you. It's those of us who contributed both to the Jepson and FMA, we're just all attempting to figure these things out. Most of us don't get paid for any of this work. It is a total work of love. But I think when somebody, in fact, just now said to me, you're a lupin expert, and I said, all I could think about is, no, I just have been studying this thing for 45 years, and I know all the questions and very few answers. But what's really important is that we have the authors of these things have taken a long look at these groups. So if you're going to study them, talk to the authors, and so that you can actually share some information. Make a good faith effort to identify plant characters that distinguish cryptic taxa from related taxa. And here's the thing, that I check the current key. If the key says banner pubescent, banner glabrous, then, look at the banner. It really isn't rocket science, it's important, just those minor things. Visit the taxon, the cryptic taxon in nature, in its field habitat, several times during the growing season. Make careful observation of the plant and its habitat. Document enough populations to determine whether the taxon is sympatric with its look-alike relatives. And, there's a lot of shrubby lupins out there. There aren't as many shrubby species as the perennial herbaceous things. But, if you've got taxa that are in the same genus, that are geographically growing in the same area, and even ecologically not separated, but even if they are ecologically separated, please, please, please look at them too. And again, ask for help from the community of practicing field botanists in the region. You know why I like this symposium is because, hey, practical field botanists, this room is filled with them. Not all of you are, and that's okay. But, it's really, it is really where we find people working, looking at the plants, whether it's Fish and Wildlife, Northern California Botanists, California Botanical Science, BLM, Forest Service, and of course CNPS. Okay, submit good herbarium vouchers to participating institutions in the Consortium of California Herbaria with complete label data. Include all characters used for determination in that plant group, describe the habitat, note plant characters that don't show or change in specimen, plant height, fragrance, color. Now, we've already said this today. I will say that the hundreds and hundreds and hundreds of specimens that I looked at for my treatments, it is so frustrating to get the label, and it had to be what I call a top snatch. So you have no idea there's a raceme, maybe, of flowers and maybe 2 leaves. You have no idea if it's a shrub or if it's an herb, and then just have, if it's an old specimen, maybe township, range, and section, and if it's a new specimen, Latin long, and that's it. You would not believe how many specimens are actually like that. So really, very important to voucher those specimens with good label data. So obviously you want today, Latin long with your GPS coordinates, you want to exactly, wherever you've taken your specimens for your study, please, submit good location data for that. I can't help it because I had so many problems with looking at specimens where, I'm trying to figure out in my key, here are some really important characters, woody shrubs versus herbaceous, height of plants, flower character, banner pubescence. So, and by the way, that little pocket, it's not just for leftover stuff. You're going to collect a lupin, collect a whole bunch of flowers and bud, and then stuff them in that pocket. Because people like me don't want to be wrecking the plant that's already glued on there. You want to wreck the stuff in the pocket. I mean, not really. But what I'm saying is that when you are collecting voucher specimens, it's really good if there's enough material out there to put the part that's in the pocket, maybe that might be a critical, component for, whether it's, an akene or whatever part. If you know your group, you know what you need to look at to differentiate them. So don't just do the inflorescence and a leaf. Don't just do fruit, no flowers. I'm always trying to look at the banner, and I'm out there with the scalpel and thinking, I know, I borrowed this and I'm sorry, scrape, but, so, Teresa, annotate all this stuff, and I'm sitting here trying to, if I could figure out where the character is on this thing, I would. So, it's very, very important to, when you are collecting this stuff and when you are noting, really, the plant is to look at those important characters. And, the key is where you find those. The diagnostic characters are actually in the key. The descriptions don't necessarily, those aren't all equally weighted in terms of what we're using to figure out what's what. Take photos of your cryptic taxa and their habitats at multiple points in their life cycle. Just don't have one like this when actually the character might be persistent racemes. And it's okay if your specimen's like this, but if you're looking at a population, it's kind of nice there on the label to say something. And by the way, that's what your collection field notebook is about. This is where you actually ramble about all the characters and things you see. And, good reading is to go back to, Jepson's field notebook and, listen to the birds as you're reading this stuff, because that's what he's talking about. So, I'm trying not to segue too much, but it's important to actually look at the whole thing, and it would help because maybe the characters in the cotyledon, in the annuals, the cotyledons are a huge character. Annual lupins, of course. If cryptic taxon has no apparent morphological traits that differentiate it from other similar taxa, make best attempts to co-author with a taxonomist that can do a morphometric analysis. This may not be your Jepson author because, like I said, we're all doing this in our spare time where we actually have a real job that pays our mortgages. But maybe you can find a student, who needs a project or whatever to actually go out there and look at some of the morphology, because when we were doing the CNPS conference, the other two speakers, they had done marvelous molecular studies, and they also did marvelous morphological studies too. And you could do both, but you do at the very least attempts to get somebody to help with the morphology. Review and compare similar taxa from multiple herbaria, including herbaria within the region where most similar taxa occur, and provide appendix and manuscript listing all the voucher specimens that you actually examine. When I was actually asked to do Flora of North America, I actually had a piece of paper that was written by Richard Spellenberger on a typewriter in 1988. I looked at that again. And I think about the tools that we have today. Oh my God, it's amazing. So that you actually can do a lot of work. You shouldn't do all the work. You need to get out in the field, but not using what people talked about in previous sessions, the digital specimens. We can look at so much more material. From my little home in the redwood forest with my slow, even DSL connection, to actually look at morphology, and you don't have to, it's actually easy to do. Lastly, if you think your new taxon is rare and needs conservation measures, write up population reports for CNDDB and or the agency that manages the land where it grows. And please provide that information during the California Rare Plant Status Review process. So, Julie, I don't know where you're sitting someplace, but on the questions, both of us can answer the questions, but I think I just want to do kind of a last plea on this subject, in that we're in a time that, I grew up in the 1970s, grew up meaning going to college in the 1970s, where everybody was talking about ecology and integration, and then things really changed in the 80s and 90s, and science is separated, and I think we need to have more of these studies where we're integrating, molecular studies with ecological studies and morphometric studies. Okay, that's the end of mine and questions.